So next we should be having Dr. Schaaf coming in to present virtually. Dr. Schaaf is the professor and chair of human genetics at Heidelberg University and the medical director of human genetics at Heidelberg University. And he is also he sits on our scientific advisory board. Dr. Schaaf has been with us from the very beginning, and I remember one of the first encounters or communication that I had with Dr. Schaaf was regarding the conference. And he reached out to us at one point, or to me, sent an email and asked to reach out to the families on Facebook if they could have their kids draw a picture and send it to him. He was putting those pictures in his lab to remind himself and all his lab workers that what they were doing was for real kids, for real people, just to bring them back to that. So that's just a little snippet of who Dr. Schaaf is, and we're very proud to have him on our team. Is Dr. Schaaf ready?
Okay, awesome. Thank you. So yes, warm welcome from Heidelberg, where the weather is ugly. It's raining and windy, and I wish I could be in Orlando with you all. But it's amazing to be connected to you all, and it's amazing to see how far you all have come since we published our first case series less than 10 years ago. And I have wonderful memories of our first family conference, which is, I guess that was also in April, so it's pretty much 4 years ago. And despite all the challenges with the COVID pandemic and so on, you made it, and you're having your conference now, and that is wonderful, because there's nothing better than being able to meet in person and being connected to other families. So I was telling about the picture which we got framed and which was up on the wall in the laboratory in Houston. And when I moved to Heidelberg, obviously the picture made it to Heidelberg with me and is hanging up on the wall in the laboratory here in Heidelberg now. Let me take that opportunity to also encourage all other families who haven't sent pictures yet to have their kids draw something and send it to me, and we'll get it framed and we'll have it hung on the wall here. And it's a constant reminder that there are actual children, actual individuals, actual families behind those molecules and behind the genetic code that we study. Science sometimes can be hard, and the students and researchers are going through situations where it's hard to find motivation because the experiments are not going the way that they want them to go. And to be able to look at those pictures and be reminded of the kids behind that disease name and the gene name is so important. So I would love to get more pictures. Now let me share my screen and get to the actual presentation. All right, you can see my screen now? Good. Today I wanted to talk about the clinical manifestations of BBSOAS across the entire lifespan, and I will present some basic information about BBSOAS and the genetics so that everybody can understand what this is all about and how we got to learn about NR2F1 and BBSOAS. But I also want to present some data from our newest questionnaire-based study in which many of you participated, which gave us a much better understanding of how BBSOAS emerges over a lifetime, and which clinical symptoms get better and which get worse, and what you can expect, especially when you have a young child with BBSOAS. That is a study that is not yet published, and we're in the process of writing up those findings, and hopefully will be published later this year. So this is where I'm at right now. This is Heidelberg, small university town in Germany. The university was founded in 1386. And this is where we have our Institute of Human Genetics. And I study rare neurodevelopmental disorders for the most part. And BBSOAS is one of our mainstays of research in the laboratory. My personal story with BBSOAS started with Peggy, who was ended up being the first patient with BBSOAS, but I didn't know at the time. Peggy was born in the same year as myself in 1978, and she had been coming to Texas Children's Hospital for decades, and she was considered a medical mystery at the time. You know, she was born with optic atrophy, and as she grew older, it became obvious that she had developmental delay and ended up having mild intellectual disability. Peggy also shows pretty significant obsessive compulsive behaviors. She was diagnosed with an autism spectrum disorder. And she now still lives with her parents at the age of 43 years in Lebanon. She lives with her parents, and every day she's picked up, goes to work, and she works in a facility or workshop for individuals with disabilities. She has a tremendous skill set. She speaks 2 languages fluently, French and Arabic, and actually speaks some English too. So quite an impressive young lady. But because of her challenges with the optic atrophy and the intellectual disability, the question emerged, you know, what could be the cause of her disease? Nobody else in her family was affected with the same symptoms. And she had had many genetic tests over the course of the years and decades, and all of them had turned out to be negative. Until in 2013, we performed exome sequencing on her. That technology had become available in the clinical diagnostic context just a couple of years before. And we did exome sequencing on her and found a misspelling in the gene NR2F1, which at that time was not considered a gene that causes a human disorder. However, we had some pieces of evidence that suggested that it might play a role. And one of those pieces of evidence was that the mutation was found to be de novo. So this is a misspelling that is only present in Peggy and not present in either her mom nor her dad. Also, when we kind of read up about NR2F1, we quickly found out that NR2F1 is a gene that plays an important role during the development of the brain, and also plays an important role during the development of the eye. So when you take that gene out of a developing animal model, there's genetic technology that allows us to take it out of the developing mouse. Those mice end up having malformations of the eyes, including disruptions of the optic nerve. So with the optic atrophy or optic nerve hypoplasia that we saw in Peggy, and having that evidence from the literature, we felt that we were on the right track and that we should keep looking for other individuals with genetic changes in NR2F1. So over the course of just a few months and working together with a group that specializes in the genetics of eye diseases in the Netherlands, we quickly identified an additional 5 individuals with NR2F1 mutations. And we published that first case series of 6 individuals with NR2F1 mutations in 2013. Since then, many more individuals have been identified and have come to our attention. Very obviously, I do not know of each and every individual worldwide with BBSOAS, but approximately 150 families have gotten in touch with us. And we keep tab and an internal registry of those individuals. So currently, as of April 2022, we're in touch with approximately 150 individuals. And I think it's a wonderful development that you are about to launch your BBSOAS registry, because that will allow us and you to get to know so much more about individuals with BBSOAS worldwide. So what are the main clinical manifestations of BBSOAS? There are some clinical symptoms that are very frequent, present in the vast majority of individuals, and those include developmental delay and the manifestations of the optic nerve. I know that Jane Edmond and V. Schaaf will present on the eye manifestations of BBSOAS a little later. So you will get to hear about, whether we consider this optic atrophy or optic nerve hypoplasia, but no matter how you put it, the optic nerves are often too small and too thin. And then also the optic projections within the brain are affected in individuals with BBSOAS, causing visual impairment. And that is present in almost 80% of all kids with BBSOAS. A lot of kids with BBSOAS have low muscle tone, and going along with the low muscle tone, oromotor dysfunction. So also kind of difficulty coordinating the process of chewing and swallowing, which can cause challenges, especially during early childhood. When we look at brain imaging of individuals with BBSOAS, we see subtle changes, in particular changes affecting the corpus callosum, which connects the right and the left hemisphere of the brain, which is often thin and malformed. Almost half of the kids with BBSOAS have seizures. Almost half of them have been diagnosed with autism spectrum disorder. And then some of them also have a diagnosis of ADHD, and about 1 in 5 children or individuals with BBSOAS has some degree of hearing impairment. Now, what causes BBSOAS? So this is a little bit of kind of like a genetics 101 course. Our bodies are made up of, trillions of cells, and within each cell, or most of the cells, there's a nucleus. And within the nucleus of the cell, we find the genetic material, and that genetic material is stored on structures that are called chromosomes. Now we have 23 pairs of chromosomes, making up 46 total, and half of those chromosomes comes from dad, and half of those chromosomes comes from mom. The NR2F1 gene is located on chromosome number 5, and mutations within one of the 2 copies of NR2F1 cause BBSOAS. We speak of a dominant disorder, and that means that the mutation of one of those 2 gene copies is sufficient to cause BBSOAS. Why are there no individuals with mutations of both copies of BBSOAS? Well, because mutations in BBSOAS are a rare event by itself. And second, we have some evidence from mouse studies that NR2F1 is so important that we expect that if you had 2 mutations, or if you had mutations affecting both copies of NR2F1, the condition would be so severe that maybe that individual would not even be viable, would not even be able to live after birth. Because if we knock out both copies of NR2F1 in the mouse, it actually causes lethality, and those mice typically don't live beyond just a couple of days of life. Now, there are different types of NR2F1 mutations, and I think this is important because each and every one of you has gotten a report that states which kind of mutation your child has. And you may want to go back to that report and look at which particular mutation your child has. First, there are chromosomal microdeletions. And that means these kids affected with chromosomal microdeletions, they have missing pieces of chromosome material. So in one of the 2 chromosomes, number 5, there's a little piece of material missing. And that piece of material includes the NR2F1 gene or parts of the NR2F1 gene, and that causes BBSOAS. These microdeletions are typically detected by chromosome microarray technology. So some of your kids have been diagnosed by sequencing technology like exome sequencing, and we'll get to those a little later. The kids who have deletions, they have been identified using chromosome microarray technology. And here on this slide, you see indicated by the black bars that deletions affecting NR2F1, they can be of different size and they can be of different location. They, like each and one of them is a little different, and you see the coordinates specified affecting that chromosomal change. And each child here is different. But the deletion always includes NR2F1. And because the deletion includes NR2F1, that affected individual manifests the features of BBSOAS. Second, there are kids with point mutations. And in fact, it's the majority of children within this group that has point mutations. These are more common than the chromosome microdeletion cases. So on the chromosomes, there are genes, and the genes are made up of a genetic code, which is basically like words in a book, and they're composed of letters or nucleotides. And the genetic code only has 4 letters. These are A, C, G, and T. And in most of the cases, the point mutations that we see in kids with BBSOAS are single base substitutions, meaning typographical errors that only affect one of those letters, one of those nucleotides. And these cannot be found by chromosome microarray technology. These cannot be found by any kind of chromosomal based technology, but you actually need to apply sequencing technology that spells out the genetic code one letter after the next. So either if you have, as a physician, if you have a strong suspicion that that child might have BBSOAS, you could sequence only the NR2F1 gene. But I expect that most of your children have actually been diagnosed by exome sequencing because the physicians were not able to piece together the symptoms. And they realized, well, this child has multiple medical issues, including developmental delay and vision impairment and maybe muscular hypotonia. I don't really know what this is. Let's do kind of like let's cast a wide net and apply exome sequencing, and subsequently the mutation in NR2F1 was found. So here in this picture, you see the NR2F1 gene on the bottom, and the black boxes mark the domains of the protein that we know are functionally very relevant. And on this graph, you see the various mutations. So each line and each combination of letters and numbers indicates a mutation that has been found in an individual with BBSOAS. And you see that a lot of the mutations cluster in this one region, which is marked as DBD, which is the so-called DNA binding domain of the NR2F1 gene and protein. And we'll get to that later. Because the type of mutation and its location actually matter, because depending on which particular mutation you have and where it's located, it in part predicts the level of severity of clinical manifestations that the child has. So in this study, we actually compared individuals who have point mutations in the DNA binding domain, marked in black, where it says DBD variants. And we compared them to children who have deletions, so children who have the entire NR2F1 gene missing. And looking at the various symptoms, you see that for several of those symptoms, the black bar is longer than the hashed one, meaning that these symptoms are more commonly seen in individuals with DBD variants compared to the deletion of the entire gene. And at that time, some of the features like seizures were actually exclusively seen among individuals with patients with DNA binding domain variants and not typically seen in kids who have deletions. That does not mean that, you know, we can predict for sure that a child with a deletion will never have seizures, but we can certainly say that seizures are much more common among kids with point mutations in the DNA binding domain. And in genetics, we call that genotype-phenotype correlations, meaning there's a correlation between the type of mutation and the specific symptoms that we see in the kids. So generally speaking, point mutations within the DNA binding domain are the most severe cases of BBSOAS. If you have point mutations in other parts of the gene, like at the very beginning of the gene or later in the so-called ligand binding domain, those again are milder, kind of comparable to the deletion cases. Now, I mentioned this earlier, BBSOAS follows a dominant inheritance pattern, meaning that the mutation of one copy of the gene is sufficient to cause disease. And the vast majority of cases, the vast majority of families we're in touch with, the affected individuals have so-called de novo mutations, meaning that the affected person is the only one in the family with BBSOAS, and neither one of the parents carries the mutation. It's a new mutation, de novo, that arose at the time that the sperm or the egg cell was formed. We are in touch with just a couple of families where the mutation was inherited from an affected parent to the affected child. And in all of those cases, the parent who carries the mutation is also clinically affected. So this shows that people with BBSOAS can go on to have children. But when they go on to have children, the chances for each and every one of their children to be affected with BBSOAS is 50%. And then there is theoretically the possibility that neither one of the parents carries the mutation, or we cannot find the mutation when we check the genetic code in their blood, but they could still have two affected children. And why is that? This is not exclusive to BBSOAS. We know this for all dominantly inherited genetic conditions. There's a chance of a so-called germ cell mosaicism, meaning that there's a subset of cells in the testicles of the dad or in the ovaries of the mom that carry the mutation. So when we test the parent's blood, we don't see the mutation, but if we did a testicular biopsy or an ovarian biopsy, we could potentially find cells that have the mutation. And in those cases, there's an increased risk or an increased chance of having another child with BBSOAS. Generally speaking, we consider that chance to be no more than 2%. So this is what we're showing here. If both parents are tested and they found to be negative in their blood and they have one affected child, the chances for having another child with BBSOAS are less than 2%. If we find the mutation in the NR2F1 gene in one of the parents, we assume that that parent is also clinically affected, and then the chances of having another child with BBSOAS would be 50%. This also means for all of your children who have BBSOAS, once they're old enough to have children on their own, for each one of their children, the chances would be 50%. Now, the important question that I wanted to get to is, how does BBSOAS evolve over time? So first, and I think it's important to state that, that in all cases, symptoms are typically present at the time of birth. NR2F1 is a gene that's important during the development of the baby, while the baby is still in the womb. So when the brain and the eyes are developing, NR2F1 plays an important role. It actually has the function of a transcription factor, which means that it affects the way that hundreds of other genes are read and processed and translated into proteins. So it does not only function all by itself and has like one specific function within the cell, but it binds to DNA and it affects how the information of other genes is read, and thereby the effects multiply, and it has consequences on multiple pathways and multiple proteins within the cell. But looking back at your children and what you've told us, it seems that very early during life, either at the time of birth or within the first weeks and months of life, it became evident that something is different about that child, comparing it to neurotypical children, and that symptoms are typically present very early on. Developmental delay is a feature that has been reported in many of your children, and especially those who have very young babies with BBSOAS. They ask, what can we expect? You know, what is the average time when kids with BBSOAS sit or walk or talk? So given the information of more than 50 families that we have gotten, it appears that the average age of sitting without support is 14 months. That's a delay compared to the general population, where we expect sitting without support anywhere between 6 and 8 months. Crawling, most of your kids crawled before their second birthday at around 20 months, but there's a wide range. You know, some of them crawl actually at 12 months. For others, it takes much longer. Some of them never crawl and they go straight to walking. The average age of walking without support is 30 months. Again, in neurotypical children, we expect walking between 12 and 15 months, and we would consider it abnormal or delayed when the child starts walking later than 18 months of age. And then that is also the age when, you know, language development happens in many of your children, and the average age of first words being spoken is also around 31 months. But we do know that we have some children who are nonverbal even at the age of like 6 or 10 years old. It seems that especially the optic manifestations are often diagnosed late. And that makes sense, you know, to some degree, because the children, you know, do not have the ability to complain or express that they have difficulties seeing. If the visual manifestations are severe enough, we may see that very early on. For example, you know, some of your kids have been reported to show nystagmus, and when a baby has nystagmus, which is like an eye movement that's typically in these cases horizontal, that prompts like an evaluation by pediatric ophthalmologists. But it's kind of shocking, and I expect Jane and maybe Vi to comment on that later. But asking you, what was the average age for the optic atrophy or optic nerve hyperplasia to be diagnosed? The average age in your children was about 5 years old. And then, you know, we've come to know that it's not just structural, but there's more to the visual impairment than just the optic nerve hyperplasia. And that cortical visual impairment plays a big role. So kind of an additional component of how visual information is processed in the brain. And that's important to understand because it affects the ways that we can approach these visual impairments by therapy. And it's important for the visual therapist to understand that cortical visual impairment plays a big role in many of your children. And the average age of diagnosis for cortical visual impairment in your children, the ones that we asked, was actually 14 years of age. So, you know, like learning more about the condition, and for us learning more as providers, but also for you as parents to learn more about it and being aware of it, you can actually take this information with you and you can alert the providers and say, hey, have you considered that my son James could have cortical visual impairment? I've been taught that this may play a role in BBSOAS children, and could you evaluate him for this? Now, do certain symptoms get better or worse over time? So what's the trajectory? This is an important question that all of you ask yourselves. What can we expect? Many of you feel like you have your hands full, and there's so many issues around BBSOAS, and you do everything to help your children, but you're worried and concerned, could this even get worse? Or is it even worth supporting my child with all these therapies? What is the trajectory of disease? So it's very important for you to understand that BBSOAS is a neurodevelopmental disorder. It is not a neurodegenerative disorder. So based on everything that we know, the NR2F1 gene and protein plays a critical role during development. It continues to play a role in like adult life and postnatal life. But we have no evidence whatsoever at this time that there's like an accumulation of like damage over the course of the years. And we have no evidence that BBSOAS is a neurodegenerative disorder. So based on what we know about the gene and protein, I would expect, and I've said this before, that what you see is what you've got, and it's a static disease that is present and will not change much over time. At least it will not get worse over time. But that was just my opinion, and so we actually asked you all, what have you seen in your children? And so in these slides, I'm showing you the individual symptoms, and I'm plotting in which percentage of children has that feature gotten worse over time, and in which percentage of children has it gotten better over time. So for seizures, you see in the vast majority of children, seizures have actually gotten better over time. Now, this could be due to two reasons. One, because children outgrow their seizures. And we actually know that the seizure threshold is lower in babies, even in the general population. And, like a lot of kids have febrile seizures, but adults typically do not have febrile seizures. So our seizure threshold as the brain develops kind of increases over time. And we do see that seizures actually decrease in kids with BBSOAS also. It could also mean that seizures are just better controlled because you have found the right medication to prevent seizures from happening. And many of those kids with BBSOAS have been on anti-epileptic drugs for a while, and then at some point, they weaned off those drugs because they don't need them anymore. Also, more generally for neurological symptoms, it seems that the control of those gets better over time, and also visual problems. Now, visual problems, I personally believe that they don't really get better because of biology, because I think the changes are there and they don't change very much over time. But I think your perception of the visual problems actually gets better because there's much that you can do with correction through glasses, etc., but also through visual therapy. You can help your children to respond better to certain visual cues and to navigate in everyday life, taking kind of the disability that they have as a challenge and to overcome some of the problems at least. Hearing problems, it's interesting to see that many of you report that hearing problems get better over time. Only a small subset feel that they've gotten worse over time, because this is something that I'm having a very close look at, and something that, we are keeping an eye on because we know from mice with NR2F1 mutations that hearing impairment gets a little worse over time. This is also the reason for me giving you the recommendation to do a hearing test in your children at least every other year, because I'm a little concerned that there could be hearing impairment emerging over the course of the years. But in fact, many of you report that hearing has gotten better over time. ADHD symptoms are better controlled over time. Autism-related symptoms, it's about a half-half situation. So these kind of social impairments, it seems that half of you feel like you've gotten a better handle on those, and the other half feels that these have actually gotten a little more pronounced as the kids have gotten older. And then there are other symptoms of BBSOAS that I haven't mentioned yet. And some of them are actually not indicators of disability, but of different ability and actually, you know, specific strengths of ability that many of your children have. So we got to realize, you know, I've talked about this for Peggy, and it has proven to be true for many of your children. They have an amazing long-term memory. And we have tested this in the mice as well, in mice where we take out a copy of the NR2F1 gene. And also the mice can remember certain things longer than their siblings without the NR2F1 mutation. So this is a positive effect. Lots of your kids have great memory. We've come to learn that many of your children love music. It calms them down, it makes them happy. And we've seen this at the last family conference, how wonderful your children respond to music. A lot of kids have high pain tolerance, which can be troublesome sometimes because they can hurt themselves and not complain about it. And then, you know, some things go unnoticed for a while because they don't complain about it. And sleeping difficulties are reported in about half of the kids, which can make your life sometimes even more challenging when the kids have big trouble kind of falling asleep and sleeping through the night. What is the age distribution in our cohort? So when I last looked, and this is about a week ago, we were in touch with 167 individuals with BBSOAS. Out of those 167, 18 were 20 years and older. The oldest individual we are currently in touch with is 50 years old. And we have a total of 4 cases that are inherited from a parent. So 4 adult individuals in our cohort that have passed on the mutation to their children. There's one single individual in our cohort, unfortunately, with BBSOAS who passed away. But this occurred at the age of 1 year, and that person actually, very unfortunately, had a second rare genetic disorder, and we believe that the impairments due to that second genetic disorder were the cause, the underlying cause of death, and that the death has nothing to do with BBSOAS. So based on what I know today, the life expectancy of BBSOAS is almost like in the general population. It's probably not quite as in the general population because epilepsy by itself is a risk for premature death because when epilepsy is severe and is uncontrolled, it can lead to death. But based on everything that I've seen and learned about your children, the life expectancy is probably very similar to the general population. So in summary, we have come to learn that individuals with BBSOAS, and I'm telling you something that you've known obviously yourself, looking at your children, they're very special people. They have several challenges and handicaps, but also they have very special abilities, and it's amazing to learn from them. Generally speaking, symptoms are almost universally present at the time of birth, and they don't change much at all over time. And if they change, they actually tend to get better, or they can improve. They can be improved by therapy. So this is really important for us to realize as parents and physicians and providers. This is our window of opportunity. We do have this window of opportunity in which we can support these children, subject them to the therapies that have proven to be helpful, and this will help them develop really to the fullest and best of their potential. And And with that, I'll close, and I know you may have questions for me, and I look forward to your questions. But before we get to the questions, thanks to all of you for allowing me to be part of your journey and allowing me and my entire team to learn from your children. Thank you. Thank you.
All right. Can you hear me? No?
I can hear you, Kali.
Oh, that's right. So, here we go. Okay. Thank you so much, Dr. Schaaf, and thank you again for, I love how you explain everything in layman's terms. You always do such a great job with that, so we can all understand it. I appreciate that. And we will get some more pictures to you soon.
Thank you.
So, anybody want to come up and ask a question? Anybody have a question for Dr. Schaaf? If you don't want to come up, you can say it out loud, and I'll repeat it in the mic.
Dr. Schaaf, I'm Maria Vessio's dad, and one of the things that we've noticed with her is with the onset of puberty, she has become very aggressive and sometimes violent and freakishly strong when she's going through this. Has anything been discovered with hormonal changes in puberty and what its effects of BBSOAS are?
Did you hear that?
So it was very faint. If you could repeat it for me,
Sure.
it'd be helpful.
We have a dad here who's asking about the onset of puberty and the hormonal changes, and how that plays a role with BBSOAS. Did I get that right? Yeah. If you know anything about that.
So it's a really good question, and this is something that we have not looked into yet. I do not have any evidence that the mutation in the NR2F1 gene would affect the onset of puberty, making it very early or very late. Obviously, we've been in touch with multiple individuals with BBSOAS who are adults, and it seems that they have fully developed kind of secondary features of going through puberty and developing normally sexually. I also understand from the female teenagers and adults that they are typically menstruating regularly, and obviously we have seen that that people with BBSOAS can have children. I have no evidence to suggest that anything would be different about them. How puberty and the hormonal kind of influx affects the features, we have not studied systematically. I mean, we see for a lot of neurodevelopmental disorders that some behavioral symptoms change, but behavioral symptoms change in neurotypical children without any diagnosis also. So sometimes aggression becomes an issue, more so in the males than the females, yeah, we do have maybe 2 or 3 individuals where psychiatric features seem to have emerged more so as the kids have gotten older and as they've started puberty. This is something that we need to keep an eye on. These are symptoms that could be suggestive of schizophrenia, like hearing voices, and these are certainly very challenging when they do occur. But at this point, it seems to me that these are isolated cases, but is something we need to keep an eye on.
Thank you, Dr. Schaaf. Any other questions? Yeah? Would you like to come up?
Yeah, I'll come up, maybe.
Great. Okay, come on up.
Hi, Dr. Schaaf. I am Teresa Grothy, mom of Logan Grothy, and he is 26, so one of the older individuals. Just wondering, I appreciated your research on the conditions that have either improved or gotten worse, or the symptomatology. Were you able to, at this point, and maybe you don't have all the data that you need, and you're asking for more of that from all of us, but were you able to make any connection with these conditions that have improved or gotten worse as it correlates with the place of the mutation or deletion?
So this is an excellent question, and I just met with my research coordinator and the physician involved in the study earlier this week, and we will be doing the statistical analysis to look at that, because you know, obviously in the past we've looked and compared the various types of mutations with the presence or absence of certain symptoms, and we've come up with this idea that there's a correlation between the type of mutation and the severity of the features. And we will now look at whether it also affects the course of disease, but I can't tell you that yet. But it's an outstanding question.
Hi, Dr. Schaaf. It's Melissa Thalen. I was hoping you could share, I know your lab is working on so much, maybe more of what you're working on, even though it's not quite ready for prime time, but some of the projects and maybe some of the grant wins that you've had there.
Yes, thank you. So in fact, yeah, my team studying BBSOAS has grown. We have some members of the team who are more involved in the clinical studies, who've been in touch with you regarding questionnaire-based studies, etc. I have one entire laboratory now at my institute that studies the basic molecular functions of NR2F1 and how it affects the regulation of gene expression in the early development of the brain, and in particular in the development of the so-called neural crest. That is the laboratory of Dr. Magdalena Lauksch, who joined my team from Cologne. And she is a very experienced scientist who does stem cell research. She works also with the iPSCs, the induced pluripotent stem cells that we generated from skin biopsies that we obtained 4 years ago. So these skin cells were reprogrammed at Baylor to become stem cells again. And we then transferred them to our lab in Heidelberg, and we use those stem cells and we differentiate them into different types of neurons. So we make brain cells or neural crest cells out of the cells that we generated from the skin cells that we obtained from your children. And these are providing very valuable insights into the molecular mechanisms playing a role in BBSOAS. Dr. Lauksch also was successful to get additional grant funding that covers a substantial research project in her lab over the course of 3 years by the German Research Foundation. Then recently we were in touch with Michelle Studer, who is a researcher in Nice in France at the INSERM Institute. And she has been studying NR2F1 for many years, even before it was known to be a human disease gene. She has a really good understanding of what NR2F1 does in the cell, and she has studied mouse models of NR2F1 for several years. And we submitted in January of this year a joint research proposal it's a transnational research call between France and Germany. We are applying for research funding to support our joint efforts to understand BBSOAS, both in mouse models, as well as induced pluripotent stem cells and organoids. So organoids are basically, I mean, sometimes people call them mini brains, where you can use the stem cells in cell culture and you grow them to develop brain-like structures in the dish. And this is something that Dr. Studer has expertise on and wants to do. And Dr. Studer is also sharing her mouse models with our lab, and we will be launching an extensive study of the various mouse models of BBSOAS because we, a doctorate student of mine, generated a mouse model that has a point mutation in the DNA binding domain, which we believe will be an even better animal model of BBSOAS. And then Dr. Studer's lab generated a mouse model with a point mutation in the ligand binding domain. So we now have mouse models with a deletion, with a mutation in the DNA binding domain, with a mutation in the ligand binding domain, and we can use them to further study BBSOAS. And we will use those to identify the very best model that recapitulates the most features of BBSOAS that we can then later hopefully also use for some therapeutic interventions. This is, maybe too early to consider the therapeutic interventions, but I know this is something that's of probably the greatest interest to all of you. So we're currently developing a research proposal, and I'll have a call with a scientist in California next week or the week after, where we will come up with an RNA-based therapeutic approach that we could probably test in our mouse models to ameliorate some of the symptoms of BBSOAS. So there's also a couple of questions in the chat online. Carly, is it okay? Should I just read out those and kind of try to answer these?
Sure, we actually have someone here moderating that as well.
Oh, perfect.
So if you don't mind, for the sake of time, can we have one or two that we'd like to ask? And then all other questions, whether if you're here in the audience or out there virtually, go ahead and if you have those questions in chat, we'll keep record of those, and any questions on HUVA. I noticed we have some questions in the HUVA app as well. We'll also take note of those, and we will do our best in the next week or so to get those to Dr. Schaaf and get those back to you, okay? I do want to say one important thing. The questions about puberty and the questions about, somebody has a question about what type of seizures manifest in kids with BBSOAS. All of these questions can be answered through the patient registry. If we all sign up and input our information, all those questions will be there. They'll be answered, and you will all have access to them. So I just have to do a little plug there about the registry. Okay, do we want to do, Brian, one question from chat, and then we'll wrap it up.
So from Nicole Groot, what type of seizures do children of BBSOAS manifest?
So there are multiple types of seizures that can manifest. The way that I see it is that generally kind of the susceptibility to seizures is greater in children with BBSOAS compared to the general population. And in part, that may be because there are some structural abnormalities or structural differences in the way that connections are formed within the brain. The most severe type of seizures that we see are infantile spasms. These can be pretty severe. They usually, as the name says, manifest during infancy, and they can be treated with rather unusual medications like ACTH, but they're important to recognize because we know that infantile spasms, when they are frequent, they can then further kind of impair the development of the developing brain. So infantile spasms are the more severe. We also have kids who have generalized epilepsy, so who have grand mal seizures occasionally when they get older. We do have kids with febrile seizures, and there's also some kids with partial seizures, where the kid is still responsive during the time of seizure, and the seizure only affects a certain part of the body.
Okay, we will wrap this up. Thank you so much again, Dr. Schaaf. Let's give a big hand. Thank you, and once again, we are so privileged to have you on our team. We will be in touch.
Thank you. Love you all. Thank you.
You.